everybody. I have 1 o'clock, so let's get started. My name is Kayla Perry. I'm an assistant professor in the Department of Entomology at Ohio State University. Today is our 4th session of Emerald Ash Borer University, which is a program that's a partnership between Purdue University and Michigan State University. My colleagues on this project are Sheila Babbitt, who is here with me moderating today, as well as Bob Bruner from Purdue. So very excited today. We are hosting Carrie Fear. Dr. Fear is an assistant professor of forest health in the Department of Forest Resources and Environmental Conservation at Virginia Tech. Her research focuses on improving forest health and resilience to invasive species using biological control, infrared spectroscopy, and silvicultural techniques. So today I'm really excited. Carrie is going to talk about a tale of 2 fungi biological control options for tree of heaven and autumn olive. So it's all yours, Carrie.
Great, thank you, Kayla. I'm going to go ahead and share my screen.
Looks great.
Thanks, everyone. I'm excited to be here today and talking to you a little bit more about some research going on in my lab here at Virginia Tech. Today, I am a forest pathologist by heart. So in this case, usually I talk about the negative forest pathogens that are affecting our forests. But today I'm going to talk, maybe give a little bit of hope on some of the good fungi that may be impacting our forests and how they can be used as biological controls for 2 different invasive weed species. So one of these we're going to talk about is biological control for tree of heaven, which is more of a biological control that's been, research has been going on for decades at this point. And then a new biological control for autumn olive that we're just beginning to research here in the lab. So 2 different areas, 2 different levels of research to hopefully give some hope for our forest. So first, I want to just start with a little bit of clarification for biological control. I think everyone is most familiar with classical biological control, which is when we introduce the natural enemy of that pest or pathogen into its non-native range. So this could be anything from like the lady beetle that was introduced here, or parasitoids that are being introduced for emerald ash borer. Very fitting. So those are our traditional, what is known as classical biological control. What I'm going to be talking to you about today is what is referred to as augmentative biological control. And this is when we are increasing the natural enemies of the pest in the area. So instead of bringing the enemy back over to the non-native range to try to control the non-native pathogen or pest, here we were actually looking for native pathogen species or native enemy species that we can actually increase in the environment and just essentially support so that we can remove our non-native pest, pathogen, or in the case we're going to be talking about today, weed species. So I'm sure we're all very familiar with Tree of Heaven, but I like this little graphic that my PhD student who is working on this project created. See if I can. There we go. To just show the devastation that Tree of Heaven causes into the environment. So Tree of Heaven is a prolific seeder and a prolific sprouter. And in particularly, whenever we try to mechanically control Tree of Heaven using traditional methods or even chemical control methods, it just comes back fiercer, more angry, and continues to reproduce sprouts, roots, and it becomes even more challenging to get rid of. This is why we see it in particularly disturbed areas. It does do better in disturbed soils and in urban areas, which is why it was originally introduced in the first place. So because of this, it's dramatically difficult to control in any settings. It can be economically challenging and then also just physically challenging as well to remove it and keep it from coming back into those areas. So a biological control option that would maintain Tree of Heaven to a that would be able to control Tree of Heaven and prevent it from resprouting would be really beneficial in terms of control efforts. And luckily for us, There was first symptoms noticed in Pennsylvania around 2002, 2003 of a wilt disease naturally occurring on Tree of Heaven in a forest. And it was later identified as showing streaking within the Tree of Heaven, indicating that it is most likely a pathogen that is causing this streaking. So there continued to be work that was done, and it was identified as a new fungal species named Verticillium non-alfalfae. And Verticillium non-alfalfae is a soil-borne wilt fungus. You may be more familiar with some of our other Verticillium species, like Verticillium dahliae, Verticillium alboatrum, and Verticillium alfalfae. For trees, we typically see these Verticillium species causing wilt on silver maples, sweet cherries, and then most common for agricultural settings is that we see Verticillium playing a huge part in disease on tomatoes and other agricultural crops. However, there was a great amount of host range testing that went on by Matt Casson at his lab at West Virginia University and by others on this project, and it was determined that Verticillium non-alfalfae luckily has no deleterious effects on any of our native tree species, which makes it a really great candidate then to be used as a biological control agent. So other areas of why Verticillium non-alfalfae has particularly been a good candidate for biological control is that it's also cosmopolitan, meaning that it can be found everywhere. So, and that doesn't even mean that it's only just found in the United States. Verticillium non-alfalfae has also been found in Europe, and they're working to make a biological control in those countries as well, where it has been found. So we don't really know exactly where Verticillium non-alfalfae is necessarily native to, but at that same time, we can find it all across the globe. So that means that we don't have the risk of spreading it into other areas, which again, really great as a biological control agent. So really, this work has been ongoing here at Virginia Tech since the early 2000s, mainly led by the forest entomologist here, Dr. Scott Salem. And at this point, it was only approved through APHIS to begin testing in states where we have actually found the fungus. So that includes Virginia, Pennsylvania, and Ohio. But it has been an ongoing effort since the early 2000s to begin testing to see if this really could be a viable biological control agent. And through the testing, it has shown that it has been very successful. So the typical symptoms that you can see are wilting in the canopy. So these trees right here in a healthy Tree of Heaven stand have been inoculated, and you can see overall the dieback that's occurring. You can also see that within this canopy photo. So overall, just a reduction in the canopy of inoculated stems and just general wilt symptoms. So at this point, we inoculate trees in May, and then we continue to monitor them throughout the season. And typically by September, we see total mortality of the trees after that initial May inoculation. And luckily, we also see it when we follow up on sites that were previously inoculated as well. So Dr. Tim Shibley was a previous PhD student working on this work, particularly looking to see what happens when we remove Tree of Heaven from the ecosystem using this biological control. And you can see the site pre-inoculation, and then 5 years later, we still continue to see tree death within the area. And luckily, with this biological control, we not only see the death of the trees where it was inoculated, but because Tree of Heaven is such a prolific seeder and a prolific sprouter, we also see that those the clonal root sprouts of our inoculated tree also then receive mortality as well, even though they weren't directly inoculated with the verticillium fungus. So again, this is a really promising biological controls that we don't have to continually retreat stands, but we could see those clonal stands of Tree of Heaven actually have mortality and perhaps even sustained mortality over time. Tim has a really great picture too that he inoculated trees on one side of a trail, and then you can see actually death on the other side of the trail following just that inoculation. So it's a really exciting tool that could be used to continue to help control Tree of Heaven. Now, unfortunately, it doesn't come without its limitations. So it does seem that verticillium has the most effect on tree of heaven when it is in cooler and more north climates. So it does seem to be very climate dependent on how effective it's going to be at actually killing tree of heaven. So there might be some limitations or dependencies on where actually we can apply this biological control. But again, it still seems like a very promising tool to add to your tool belt for invasive species management. So the next steps of this work are really to see if how we can make it actually applicable for land managers and other people to actually apply it so that we can begin to control tree of heaven in the ecosystem. Because up until this point, what we have been doing is testing inoculation methods, really doing a fungal dilution and then dropping it into the tree using a gouge and dropper method. And essentially the tree, the xylem uptakes the fungus and then it is systemically transported throughout the tree. But of course, if we want to actually make this something that can be usable by land managers, this would not be the most feasible way for us to apply it or direct other folks to actually use it. So a question that we sought out to answer is, can we develop a commercial formulation of this verticillium non-alfalfae that can be used by land managers? So to do this, I am part of a multi-state, multi-university and government team funded by a USDA Agricultural Research Station area-wide grant, focusing on using biological control to manage both spotted tree of heaven and then its native predator, I'm going to call it that, of spotted lanternfly. So through this grant, we have been looking at not only the impacts of verticillium non-alfalfae on tree of heaven, but also if there's potential biological control agents, particularly Bovaria bassiana, another soil-dwelling fungus, to control spotted lanternfly. And then also, what happens when we actually inoculate tree of heaven? What happens to spotted lanternfly when we have sick trees within the area? So this is all part of this larger grant, particularly focused on vineyard areas and how we can actually make verticillium non-alfalfae commercially available to these owners of vineyards where spotted lanternfly would be most likely to be the largest problem. So in order to develop this commercial formulation of verticillium, we luckily are able to partner with Bioproducts Inc. and their CEO, Charu, who is a former faculty member at University of Florida, and then started this company in order to try to produce biological controls of fungi. So with collaboration from Bioproducts, we developed a powdered formulation of the verticillium non-alfalfae in order to begin testing to see the efficacy of our lab formulation method and then this commercial formulation method. So my PhD student, Harrison Miles, has been tasked with, one of his tasks has been actually testing to see if how effective is the commercial formulation compared to the lab formulation. So in the summer of 2025, he established 6 treatment plots at 2 different sites. So there were 15 plots in total, with 10 trees per plot that were inoculated with either the lab formulation, so this verticillium non-alfalfae 140, or the commercial formulation of the Sylvan 3. To see again if we saw any differences between the 2 different formulations to cause disease on tree of heaven. And based on the area of the disease progress curve or the number of trees and their symptom severity over the summer of last year, we saw no significant differences between our commercial formulation and the lab formulation of verticillium non-alfalfae, which is really exciting, meaning that we can continue to develop verticillium non-alfalfae as a commercial formulation and be able to actually get it out to landowners in order to try using it. So the next steps, particularly for this side of the research, are now to test more effective field options. So of course, we have been using that gouge and dropper method. Again, not going to be a feasible field applicable method for land managers. So the goal would be to test methods that would be able to be used by methods, like land managers. So this could include using hack and squirt or looking at basal bark application methods. So other ways that we can actually see the efficacy compared to that gouge and dropper method. And then the next part is to determine Verticillium non-alfalfae's effects on spotted lanternfly. So we now know that Verticillium is effective for controlling tree of heaven. But a question is, now that we have spotted lanternfly in the area feeding on tree of heaven, is it possible that spotted lanternfly could actually pick up Verticillium and then transmit it to neighboring trees? So this is something that's also ongoing in the lab to see if it is possible to actually have it be vectored by spotted lanternfly. In addition, what we're also looking at here is to see if Verticillium non-alfalfae has any effects on the fitness of spotted lanternfly as well. So the hope would be that we can use this fungus perhaps to control 2 invasive species within the area. So switching gears a little bit now to another problematic invasive weed species, which is autumn olive. So again, I'm sure many of you are already familiar with autumn olive and maybe its infamous issues that is associated with it. But just to give a little background, so autumn olive is native to Asia, and it was introduced to the United States in the 1830s to provide cover for disturbed sites. It forms dense patches in the understory and is has a nitrogen fixer, so it is very competitive with other plants. Its berries are produced into the winter and provide a great food source, especially for birds and other animals. And similar to tree of heaven, it is a prolific seeder as well. So we can have those seeds be spread very far based on birds and other animals as well. And like tree of heaven, it is very hard to control. So it becomes, again, forms these dense thickets. It can be very challenging to remove it because there's thorns that are produced from it. And you have to either completely remove the root system or apply several applications of herbicide to keep it from growing back and from resprouting. So it can be quite challenging to remove, especially an area that's heavily populated by autumn olive. So, and to this point, there has never been a biological control that has been successful at actually controlling autumn olive. Typically, the applications to do any of the control are basal bark applications using herbicides. However, in the spring of 2024, some of my colleagues were out at the Powell River mine site, which is a mine reclamation site here in southwestern Virginia. And they started to notice large mortality of these mature autumn olive. And upon closer investigation, they noticed xylem streaking within the plants, large cankers, and then just overall dieback of many of these autumn olives. So they approached me about it to ask if we had ever seen a fungus that was impacting autumn olive or what pathogens might be impacting it. And I wasn't familiar with anything that was actually causing disease on autumn olive. So that led to a seed grant through the Powell River Project Reclamation funding with Sarah Clough, who is the one who actually saw the mortality occurring at the Powell River Project. Dr. Jacob Barney, he is the director of the Invasive Species Collaborative here at Virginia Tech. And then 2 additional researchers, Dr. Dan Putnam and Patricia Donovan, who work with remote sensing and UAV monitoring. And through this project, we wanted to, 1, determine if there was a species of fungus actually impacting autumn olive and what that species was, but then to further determine the extent of the infection at the Powell River Project. For a little bit of background, the Powell River Project it still has areas of active land management or active mining going on, but then is primarily being restored using different forestry techniques. But at the time when it was first becoming reclaimed, there were recommendations to plant with autumn olive, which is why so much of the cover is dominated by autumn olive that are mature, thick-stemmed, and outcompeting many of the native vegetation in this disturbed area. So we wanted to see the extent of the area and how it's impacting other reclamation techniques that are already ongoing at the Powell River Project. So to do this, it was done under the work of two undergraduate researchers, Helena Czar and Jacob Knuff, and then a master's student of mine, Shane Allen. And essentially, they were all tasked with determining what, if any, fungi were present within the diseased autumn olive. So this involved fulfilling Koch's postulates, so determining pathogenicity of any of the fungi that were isolated. So these students isolated what were considered three morphologically distinct isolates from these infected autumn olive. So those isolates, how we're naming them, are E3I1, H3I3, and H3I1. So all morphologically distinct, or what we thought were morphologically distinct. But I did want to point out to all of you that you can see that there are some large overwintering structures that are present with each one of them, which to me would suggest that perhaps these are all part of the same genus, or at least the same family of fungi, and very distinct of a certain pathogen group. So with these three distinct morphological fungi identified, we then performed a PCR and sent away for sequencing, and then performed phylogenetic analysis on each one of these to actually see if we could get to the species level of each of these fungi. And luckily, we were able to get to the species level. So the E3I1 was identified as a species in the Diaportha genus, Diaportha ellipsoidea, while the H3I3 and H3I1 isolates were all determined to be of the species Diaportha aries. And Diaportha is very characteristic of these large overwintering structures that we're seeing here. So far morphologically, this would make sense that we're seeing these distinct species types with the fungi that we extracted. So let me give you a little background of Diaportha here. So Diaportha was previously known primarily as Thamopsis, that is the asexual state of the fungus, until it was renamed to only go by the sexual states of the fungi. So you may be familiar with Thamopsis stem canker or blight. This is very impactful on black locust, American sycamore, and also on black cherry. And it is also, Diaportha is commonly a huge problem in agricultural vegetation such as tomato. But for the most part, we consider it an opportunistic fungus. That means that it only impacts trees or plants that are stressed or have wounds and openings that allow Diaportha to enter and cause disease. And this actually makes sense for what we were seeing at the Powell River Project. So this picture here is showing a black locust species that is heavily cankered with similar symptoms that we were seeing at the Powell River on our autumn olive, which would suggest that perhaps there is Diaportha within the environment causing infections on other species besides just autumn olive. It is also considered cosmopolitan in some respects, which is why I have the question mark here. There really isn't a lot of information on Diaportha species in particular. So it's very challenging to get to the species level identification of Diaportha because they are so similar, which is why they tend to be thought of as a species complex. And even that Diaportha aries species is usually considered a complex of several different species that are able to cause infection. So this cosmopolitan I have in a question mark because we do see different species of Diaportha across the United States and very common in forests and other agricultural settings, but it's not specific to just the species that we found. At this point, it is unknown or there are no reports of it impacting autumn olive. There is Diaportha species recorded on European autumn, and Diaportha aries in particular has been detected on juglans species as well as fruit trees like peach trees and other species in orchards. So it is not uncommon to see Diaportha infecting trees, but right now it has never been reported on autumn olive, which makes this be the first report of it. It is also a well-documented to cause disease and wilt on Russian olive as well. So again, making it perhaps a primary candidate for biological control, or again, not uncharacteristic that we would be seeing Diaportha on autumn olive in these areas. So now that we have our morphologically distinct isolates actually isolized and then identified to the species level, we decided to do a greenhouse trial to see the true effect of these fungi and actually try to prove Cook's postulates, to try to prove the pathogenicity. So we had 4 treatments, so each of our single isolates. And then we also decided to do a combination of all 3 of the isolates because it is very common, like I said, for Diaportha to be more like a complex. So you might, in order to cause disease, Diaportha may have to interact with one another to actually cause disease symptoms. And since we isolated all 3 of these from symptomatic plants, it's possible that they have to work together in order to cause symptoms. So we tried those 4 different treatments as well as a control. And we had 3 replicates of each for a total of 15 plants. And we allowed the fungus to try to cause disease for about 12 weeks. And to do this actual inoculation, we took phloem plugs directly from our Petri dish of the growing fungus and created a wound on the plant and then placed the fungus onto the plant and wrapped it with parafilm to create an environment for it. And then again, checked them every other day to record disease symptoms such as wilting, cankering, and then at the end of the trial, we also wanted to see if there was any streaking present within the trees. So we checked on the symptoms every other day for those 12 weeks. Now, unfortunately for us, we didn't see any total mortality at the end of those 12 weeks. However, we did see wilting and symptoms being produced, but there was no total death of the trees within the greenhouse. However, when we were taking samples to then try to re-isolate the pathogens, we did notice that several of our trees had significant streaking within them, so both above and below the area of inoculation. And then more so, we actually saw the fungus was beginning to produce overwintering structures and also cause more disease and sunken lesions above and below the inoculation point. We also saw some epicormic sprouting begin to produce as well, indicating stress on our autumn olive. So even though we didn't see total mortality, we were beginning to see some symptoms on the autumn olive. But much to this invasive species testament, it was very hard for us to even create conditions that were not good for the autumn olive. So they were thriving in our greenhouse, just doing great, which made it interesting to think about what this fungus might need in order to cause full infection. Because in this controlled setting, the trees were doing quite well. So that might be one of the reasons why we didn't see total mortality. But we still wanted to explore, okay, was there any differences between the symptom types that we were seeing between treatment types? And what we saw is that our H3I1 isolate, which is the Diaportha aries, was significantly different than the control, as but was not significantly different from the H3I3, which is the other Diaporthes aries isolate. So this could potentially mean that we are dealing with two different strains of Diaportha aries, or again, that these have to be greater separation by greater and deeper phylogenetic analysis to understand if it really is one species of Diaportha aries that we're dealing with, or if they're just two closely related species and we need to do some more phylogenetic differentiation to see what these two species might be. But overall, we did at least see one treatment that was significantly different from our control. We saw no differences with the combination treatment, so with the multiple morphologically similar fungi together. We didn't see any differences from the negative control with that, leading to believe that at least one of these fungi is more virulent in the ecosystem to cause infection. Now, as I mentioned, we didn't see actually any deaths within the greenhouse, which led us to some additional considerations is, does autumn olive need to be predisposed to stress to actually cause mortality? So where we were seeing this fungus in the landscape was on a reclaimed mineland site. So the soil has been is very degraded. There's not a lot of diversity. We have thick stems. We have invasives present with all over there. So it's really poor conditions. And although autumn olive tends to do really well in these conditions, it doesn't always like shade. And at this point, because autumn olive was so large, it was beginning to shade and even outcompete one another. So it is the potential that we are seeing stressed autumn olive at this site, and then leading to further mortality from the Diaportha species. So it's possible that actually it's not the Diaportha alone that is responsible for its death, but that autumn olive needs to be stressed first, then see infection, leading back to that opportunistic nature of Diaportha. Now, exciting for us, this seems to also be true because we were seeing similar symptoms when we started to do this research near a park in Blacksburg that was where we saw autumn olive that was being totally shaded by larger canopy trees, which would lead us to, again, believe or further support the idea that autumn olive needs to be stressed in some way in order for us to see these symptoms. So we took some of this back to the lab and also confirmed that this was Diaportha that we were seeing in Blacksburg affecting our trees. So we're doing further validation to see, again, if this is the same species that we're seeing or if it's another Diaportha species that may also be able to just impact autumn olive throughout the different areas and counties of Virginia. So with this in mind, and this hypothesis that perhaps we need to have stress on our trees to actually cause infection, we decided to also do a field inoculation of our Diaportha species. So set up in the same way that we were doing our greenhouse trials, we tried our virulent strains, so the ones that were identified as Diaportha aries, the one was diagnosed or identified as Diaportha ellipsoidea, and then combinations as well as controls in 2 different sites with 3 plots per site throughout the Southwest Virginia to test to see how this actually responds to mature autumn olive in a variety of conditions. So we did this inoculation actually in the fall of last year. And why we did that in the fall is because we also wanted to know if perhaps Diaportha aries needs a time of overwintering in order to cause infection. So this was also something different that was in the greenhouse, was when we inoculated was during the growing season. So the fungus didn't have any time to go through dormant period and create overwintering structures. And we actually saw a similar thing happen when we were working with Verticillium non-alfalfae. That Verticillium actually has melanizing structures within it. And those melanizing structures allow it to be more virulent and then inoculated into Tree of Heaven. So we wanted to see if there had to be a similar effect with Diaportha, so we chose to inoculate in the fall, and we are now just starting disease checks in the spring. So far, we haven't seen any outright mortality from that overwintering. This was just right after leaf out of autumn olive. So it appears that if this truly is a virulent fungus that can impact Diaportha, that we wouldn't see it immediately in the beginning of the growing season. But some other questions that could result from this is maybe it's not the best idea to inoculate in the fall. Perhaps it really does need to be inoculated during the growing season. So additional questions that we need to answer at that point. So the next steps here with Diaportha are to construct the host range of Diaportha aries and even Diaportha ellipsoidea. So although Diaportha may be a cosmopolitan species, or we're already finding several other species of Diaportha within the areas impacting autumn olive, what can we expect for it to impact? And that is anyways what we need to do for APHIS in order to get permits to test this as a biological control outside of where we're finding it, is to actually determine what the host range of Diaportha aries is. The second step would be to assess the virulence of autumn olive across a range of environmental gradients and host characteristics. So this is similar to what we were doing with Verticillium non-alfalfae on Tree of Heaven. So again, we found that Verticillium did better in cooler, more north climates. And again, we could perhaps be seeing something like this with Diaportha aries. It's possible that either the host, autumn olive, needs to be stressed in order to cause infection, or it's potential that there has to be an area where Diaportha aries can thrive in order to be more virulent and actually cause disease. So we need to test that to actually assess how effective this pathogen may be at controlling autumn olive throughout its range. And then much like what we're trying to do with Verticillium non-alfalfae, we need to evaluate the most effective and feasible application for Diaportha aries as a biological control for autumn olive. So this would, again, see if we can develop non-lab methods. We're still using that PDA method when we're putting it directly into the tree. But is there some other methods that would be more applicable for our application methods, whether this be basal bark application, or if we find out that actually the autumn olive needs to be stressed in order to cause disease, we have been considering if there's a way that we can first inoculate or inject the tree with an herbicide and then follow it up with Diaporthas to actually cause total death of the autumn olive, if that would make it more effective on a shorter term timescale. There's also some consideration too that even if we can't use this as a biological control, are there cultural methods that we can use in an environment where we find Diaportha aries to promote infection? So perhaps there's ways that we can create a more humid or a more cool climate, or put particular species within the area that would impact Diaportha aries and make it more virulent within that area. Or if we just simply cause wounding to the autumn olive, well, that may make it more effective for these natural infections to help cause mortality. So there's several ways that this new fungus maybe could be used and be effective within a certain range to actually cause mortality or infection of autumn olive. I don't think either of these, Verticillium non-alfalfae or Diaportha, are going to be the silver bullet to controlling the invasive species. But again, if we can create a sustainable level of the pathogen within the environment, it can help restrict it from coming back, or and the way that it has with previous management techniques. So I think it could be a really effective tool in both of these cases to help land managers and help prevent it from spreading to other areas where we would not want it to spread. It could be an economically and a more ecologically sound method from if we do the testing that would be appropriate to employ both of these biological controls. I think I ended with plenty of time for questions, and I'd be happy to take them all. But thank you for your attention and for inviting me to this course.
Awesome. Thanks so much, Carrie. That was great. Folks can add their question to the chat and I will read them off. Looks like we have our first question regarding host specificity of V non-alfalfae. Very glad to hear of the lack of impacts on native tree species. Are there any known impacts on other non-native plants other than tree of heaven at the moment, or is there reason to expect that other hosts could be found?
Yes, that's a great question. So it's funny, actually, that's how we can even thought of this idea for the autumn olive. Sarah Clough approached me and she's like, oh, could this be verticillium actually impacting the autumn olive, similar to what we're seeing with Tree of Heaven? At this point, we haven't seen its infection on any other non-native plants yet, but it also hasn't been something that has been directly done either. So it could be the hope that we can continue to see it, but at this point, it seems to be very specific for Tree of Heaven.
I guess as a follow-up to that question, so I am really interested in the idea that you're investigating whether spotted lanternfly can transmit. So I guess following that like same line of evidence, like there's no potential then, like if spotted lanternfly can transmit verticillium, that it will be affecting other plants that it's feeding on.
Right. So that is a great question. So as of right now, no, there wouldn't be any of that evidence just based on the host range testing that we already did. So even with the way that spotted lanternfly feeds on the plants, it's probably unlikely that it's going to really pick it up and then continue to move it. But as of right now, no, we've even tested verticillium on grape as well, because we plan to employ it in vineyard settings to the Tree of Heaven surrounding the vineyards. So of course, that's a huge management concern that if we're putting verticillium in vineyards, what's going to happen? And we've seen no effect on grapes. So with spotted lanternfly, it doesn't seem to be an issue. Interestingly enough, the other biological control agent that is still in quarantine for Tree of Heaven is the Ailanthus trunk weevil. That's been really interesting. So we've done some testing already with that biological control, and it is able to pick up verticillium and move it into other Tree of Heaven. Now that feeds very differently than spotted lanternfly. But if we're able to get APHIS approval for the Ailanthus trunk weevil to be released, that does seem to be a promising one that could move the fungus. But yeah, the verticillium still has some questions, particularly with spotted lanternfly to answer.
Mm-hmm. It's very interesting. Any other questions? I have a bunch, so but I'll wait. How long have you had spotted lanternfly in the area, and have you seen, and maybe you haven't directly like looked at this, but even anecdotally, are there any like synergistic or additive effects on Tree of Heaven from verticillium and spotted lanternfly together?
Yes, so our quarantine for spotted lanternfly was just lifted last year throughout the state. We saw really our first adult spotted lanternfly last summer as well. So very recent. It's been in Virginia though since I think at least 2021, but just more in Northern Virginia. But so at one of the field sites where a PhD student Harrison is testing, it has been really interesting to see that spotted lanternfly, at least the nymphal stage so far, tends to avoid infected Tree of Heaven, which is interesting. So there might either be some sort of a less attractiveness of verticillium infected trees that spotted lanternfly doesn't seem to want to feed on. So we're actually conducting those trials right now, one to do a preference and then also do a fitness trial to see what spotted lanternfly tends to do with infected Tree of Heaven. Anecdotally, Harrison is noting that it tends to move up the tree. so that it's infecting more of the new branches or the new leaf out, as opposed to wanting to sit on the trunk where it was infected with verticillium. So really interesting. And then going back to the vineyard, we're curious if it's the hope that removing Tree of Heaven would just remove spotted lanternfly, but that's not exactly what we've seen in Pennsylvania. So we also don't want to be removing Tree of Heaven to then get spotted lanternfly to move further into the vineyard. So it'll be interesting to see how spotted lanternfly behaves with verticillium.
Yeah, that is interesting. I don't know if I would have predicted that they would avoid those trees, but yeah,
Yeah.
I don't know much about diseases, so maybe that's why. Yes. Any other questions? I do have one more and then I'll stop, I promise.
Oh, no
Um,
problem.
for your autumn olive experiment where you were testing the different isolates of Diaportha, I found it interesting that the combination wasn't different from the control. And even like if that one, the Aries isolate was in there, like it wasn't having an effect. And I guess uh, like are they known to inhibit each other or what might explain that happening, like that
Yeah,
observation?
that was really interesting to us as well. I predicted that was going to be the most effective. I thought that there would be enough of it within there that is just multiple species in there causing actually the infection. However, it doesn't, yeah, it seems like there might be an inhibition effect that's going on with one another. So we also, our tree ecophysiologist here, he actually did some stomatoconductance as well to see if he could see any changes. The lowest stomatoconductance was again with that only the Diaportha aries, but we saw no difference with the combination between the control there as well. So yeah, that absolutely warrants some further investigation if there might be some inhibition that they're doing. But typically they act together, at least that's what we reported in the literature for Diaportha, that they do better together to cause infection. But
Mhm.
perhaps that isn't the case, at least with the species that we found.
Yeah, did you find like a combination of all three isolates in the field when you were like originally isolating them? Like,
We did, yes. So we found a combination of them from multiple stems that weren't infected. So either they're being infected in different areas of the plant, or it's that one is acting more as a saprophyte, so not causing the infection, but just living on the tree, and one is acting more as a pathogen. But we are certainly
hmm,
finding them. It could also play too that we're finding different species of trees that are infected with the Diaportha. So there could be perhaps some that have just been in the area and are just living on. We didn't surface sterilize any of the material directly because we wanted to see what was maybe going on inside of them. So it could have just been that we picked something up on the end, but it was enough that we could get them to grow, which is interesting.
yeah.
It wasn't just like environmental DNA.
Hmm. I wonder if there's a temporal component too, where like the pattern of colonization is important, like who colonizes first, and if there's like some priority effects there.
Yes, yeah, yeah, absolutely. I think the environment is going to be really the next interesting step to see
Yeah,
what that tells us.
yeah, that's awesome. I'm really looking forward to seeing where these projects go. Super
Yes,
interesting, even for an entomologist, where I don't mess with bacteria or pathogens at all, but it is fascinating.
yes, well, thank you.
Yes. Any final questions for Carrie? All right. Well, thank you so much for being here today and sharing your work. This will be recorded, and so it'll be up on the YouTube channel, and you can track views over time for extension and stuff, which is great. We do have our next session on th Thursday, June 4th, where we'll have Olivia Bigham from Purdue talking about her research. So thanks, everybody. Have a great rest of your day.